My name is Dušan Veličković, and I work here in EMSL as a chemist. And today we heard a lot in the previous talks about how technique called mass spectrometry imaging, particularly MALDI mass spectrometry imaging, can be integrated in different workflows to answer different environmental questions. So today I actually want to go into some fundamentals of this technique, especially of the MALDI mass spectrometry imaging, and to show you also some additional examples how this will be integrated with other omics and imaging approaches, but also what we can learn from this technique if we use it alone as a technique. But also I want to hint on some limitation of especially metabolic imaging using mass spectrometry imaging, and then how here in EMSL we developed some advanced platforms that can bypass those challenging, particularly on-target chemical derivatization workflows, and then how we apply that in some of the platforms that we heard today, especially terraforms, but also another one for the rhizomap, actually, to see molecular distribution inside the rhizosphere, actually. So mass spectrometry imaging, for those who are not familiar with this, is actually spatial probing of sample surface chemistry by a mass spectrometer. So what we are doing here, actually, as you can see on the figure, we go from the position to position in the sample, from pixel to pixel, and acquire mass spectrum, where mass spectrum actually reflects molecular composition in that particular area of the sample. And then when we finish our entire scanning of the sample, we ended up with those ion images that actually represent a relative abundance of molecules across the surface of the sample. In this case, this is root tip. So at such in that manner, this provide you spatial distribution of molecules. It can be completely untargeted or a targeted approach. So it can be completely exploratory, or we can just follow some specific chemical and their kinetics and dynamics in the sample. And actually, mass spectrometry imaging can be used for analyzing molecules in the big range, from the small molecules, like, for example, pyruvic acid, up to the small proteins, actually. And then depending what we use for the sample probing, which is usually like either like kind of laser or maybe micro liquid microjunction, we can have like different spatial resolutions here, so we can analyze, for example, clusters of cell in terms of tissue functional units, how we like to say, all the way down to the single cell level, even subcellular level, depending, of course, how well we can focus our probe. And EMSL here, Tamas mentioned that at the beginning, is equipped with really a lot of tools that enable analysis of environmental processes at different scales. But today I want to focus on the MALDI mass spectrometry imaging. It's the most common, most robust, and definitely most employed mass spectrometry imaging modalities for analyzing of intact molecules, so for molecular analysis. And as you can see from the name stands for the matrix assist laser desorption ionization. That means that this technique requires so-called application of the MALDI matrix. That is just, you can imagine, a small aromatic acid or base that will actually absorb UV energy of the laser that we use for the probing of the sample, and transfer part of that energy to the analytes to ionize them, because in the mass spectrometry, what we measure mass to charge ratio. So we need to convert molecules to the ions, and then depending what MALDI matrix we use, we can choose what type of molecules we will analyze. So we will not use the same MALDI matrix if we want to use, for example, small organic acids, or if we want to use and analyze like peptides or for example, glycans, etc. Also in MALDI mass spectrometry imaging, optimal resolution, the resolution that we use here is roughly 25 microns. Some other instruments we have that we have right now can go even like lower, like 5 micrometers. And optimal mass range usually is in the commercial ones instrument, in the one that we use here, FTICR, from 100 to 2000 m/z. But we also have other instruments that I will not cover today, like OHMR, that can cover and analyze, for example, small proteins that are up to, for example, 20 kilodaltons in size. So mass spectrometry imaging as standalone techniques can answer many biological questions. Here I put just some example. If we want to, for example, observe metabolic translocation in the roots during interaction with microbes, or to create 3D metabolic maps of plant tissue, like in this case, soybean root nodule, or when we want to investigate spatiotemporal metabolic responses to drought stress in leaves, we can also visualize metabolic topography of microbial colonies, or investigate microbial or metabolic exchange on microbial interface. So basically, we use MALDI mass spectrometry imaging to have like kind of snapshot of molecular distribution in that particular moment. However, if we combine mass spectrometry imaging with some other imaging or omics workflows, we can provide a more complete picture of some process. And we here today really great example from Jocelyn, Runima, Odetta, as well, how MALDI mass spectrometry imaging can help answer some of your questions, especially for that, from that molecular and spatial aspect. Here is another example that I want to highlight here, developed by Maria and Christine Burnham Johnson, that called MIPI workflow, where basically we use MALDI mass spectrometry imaging to capture spatial metabolism, or capture molecules. In this case, this is a fungal garden sample, and then use exactly same sample to capture protein abundance in those areas where we capture the metabolites, using also capability developed at PNL that calls nanopods. And in that way, we can basically correlate substrates and products with enzymatic activity, actually with the enzymatic abundance in that particular area, what can actually, in this particular case, allow us to map actually lignocellulose and better describe lignocellulose decomposition by microbial consortium in those fungal garden samples. Another example that I want to show this is work actually of Malak, and she will have talk, I think, tomorrow. So maybe she will cover some of this, is how we can combine MALDI mass spectrometry imaging with, for example, NMR and nanosims to elucidate drought tolerance mechanism in plant roots. So here I want to highlight that each of those techniques has different output. For example, NMR will give us exact metabolites ID. However, we will lose spatial information there. But then when we use MALDI mass spectrometry imaging, we can reveal exact metabolite localization at cellular scale. But also we suffer from exact metabolite ID, because here we measure molecular formulas of the compounds. So some of the many compounds, we call them mass isomers, can have like same molecular formula but different structure. So in that sense, we are limited in MALDI mass spectrometry imaging on exact molecular formula, but not necessary on the exact metabolite ID. And then we can use nanosims that can go even subcellular, in the subcellular domain, in the subcellular resolution. And we can reveal, for example, where C13 carbon is enriched, because at the beginning of the experiment, plants were fed with C13 pyruvate. So we can reveal where that C13 enrichment went in the subcellular and cellular scale, and then connect that to the molecular formula and spatial localization that we revealed from the MALDI mass spectrometry imaging, and then again correlate there, and from there reveal what is exact metabolite ID that got enriched, and in what what cells, in what type of the cells in this particular root cross section. However, I want to talk here that MALDI mass spectrometry imaging cannot resolve everything. So there are some general limitations of this technique. In the first place, we cannot really be sensitive to low abundant molecules like phytohormones, for example. Also, poorly ionizable molecules. Remember that if we want to do mass spectrometry imaging, we need to convert molecules into ions. So those ones that are poorly ionizable are quite challenging to detect. And then because of instruments constraints, molecules that are very low in the mass, like below 100 Daltons, are also undetectable in our capability. So also we have other problems. Some of them I already hinted before, that isomeric species, those that have like same molecular formula, so same mass, are undistinguishable. But also we have problems in confident molecular identification, because in the lot of, especially if we go in the low abundant molecules, C13 isotopologs, and here we use isotopic fine matching for molecular conformation. Those C13 isotopologs can be really often below limit of detection, which actually decrease our confidence in accurate molecular formula identification. And in EMSL here, in the work with Kevin and Vivian, we developed a workflow that bypass many of those challenges, and it is based on the on-target chemical derivatization that basically we introduce some chemical compound that has a permanent positive charge, and then we derivatize carbonyl compounds, mostly aldehydes, ketones, and carboxylic acids. So we boost their sensitivity in terms of ionization, of course, but also because we added tag to the molecule, we're pushing small metabolites toward higher masses that are optimal for our mass spectrometers. And then also because chemistry of derivatization is different. If we have aldehydes or ketones or carboxylic acids, then we can even distinguish isomeric species that are composed from those functional groups. So this is example how, if you see here, actually this derivatization compound contain a bromine atom in its structure. So when we introduce this in our analyte, we have those two stable bromine isotopes 79 and 81 that are very similar in abundance, and that facilitate our annotation workflow or framework behind it to recognize those patterns and to confidently ascribe and say, okay, this molecule is derivatized, this is not, and this is exact molecular formula of that particular compound. So I want to show you now how we actually applied and where we applied this on-target chemical derivatization workflow in several examples. At the first place, to reveal some acidic compounds in microbial interaction, and then how we can use this to visualize root intercellular metabolism, but also extracellular metabolism inside the rhizosphere, on the example of the rhizochip as a synthetic soil environment, and our newly developed platform called Rhizomap that use native soil environment, actually. So, didn't advance. Oh, yeah. So basically, we can just place the microbes on the agar plate and use this on-target chemical derivatization workflow coupled to the MALDI mass spectrometry imaging to reveal distribution of acidic compounds directly from those colonies that are grown on the agar. And this is not possible using traditional MALDI mass spectrometry imaging approaches. In the first place, to don't go in the mechanistical details, but because if you want to analyze acidic compounds, you need to use like negative ion mode analysis, and it is not compatible with the agar surface. So this is first time that we were actually able to reveal distribution of free fatty acid components of TCA cycle and other carbonyl containing compounds in different zones on interaction between the Bacillus subtilus and Fusarium. Then in terms of intercellular metabolism, we always need to cut and open cells to reveal what is inside. So this is example on the cross section of the poplar root, how we can image entire TCA cycle or phytohormones in the poplar root, or some biologically active aldehydes, for example, in the soybean root nodule. Then we can also do and integrate this or overlay this with the optical microscopy to reveal exact cellular specificity of those compounds. Like in the case of the poplar root, for example, you can see that this stress reporting compound methyl glyoxal is present in the vascular cells of the poplar root. And then you can see also how, for example, aspartate is more abundant in the drought when we expose poplar to the drought stress, and how it is localized in the root tip. And then I can skip this slide because Arunima already introduced this platform, but we made it actually compatible with the mass spec imaging, where we can investigate root exudation in those synthetic soil habitats. And this slide or similar, Arunima also showed where we can see how different type of molecules from amino acids, components of TCA cycle, free fatty acids, etc., are present in different types of the roots in different conditions between the channels of the pores, etc. And also we can zoom even in and visualize some hotspot of metabolic activity by using high spatial resolution MALDI mass spectrometry imaging. And finally, I want to show you how we can actually use and investigate root exudation in the native soil environment, basically by growing plants in the rhizoboxes. And then we can basically place the membranes or some kind of imprinting materials between the plexiglass and put it membranes on the root soil interface, imprint, so transfer molecules to those membranes, and then do mass spectrometry imaging, basically placing the membranes on the MALDI target plate, perform on-target chemical derivatization, MALDI matrix application, and shoot each position of the membrane with a laser. And you can see here some of the results that we can obtain here, how we can map and provide metabolic view through the entire depth of the rhizosphere. So from the root base to the root tip, and some molecules we can detect 300 to 400 of metabolites in using this approach. But you can see how, for example, pyruvic acid, methyl glyoxal, glyoxalate, glyoxal as well, how they kind of co-localize with the root, but also diffuse in the surrounding soil channels and soil pores along entire root length. But for example, you can see that oxoprolin actually has gradient of abundances, where it is most abundant in the root base. And then we have like this on the first and the first image is secondary metabolite, the cucurbitacin, I think it's pronounced good, and it is actually like localized exactly on the epidermis of the root. It doesn't exude in the surrounding material, in the surrounding soil and medium. With that, I would like to finish this talk, to thank all Pacific Northwest National Laboratory people that contributed to developing this imaging or MALDI mass spectrometry imaging workflows, and of course the funding EMSL at the first place. So